Although there are a lot, but not enough, antibiotics, they tend to target a core set of bacterial processes as well as their structures. And so speaking of structures, some of the main antibiotics you might know about, things like beta-lactams, things like penicillin and ampicillin, as well as glycopeptides, things like vancomycin. These target the making of the bacterial cell wall. We don't have a cell wall, so great target, right? Problem is, bacteria can evolve resistance by taking those beta-lactams, say, and making, well, a beta-lactamase, so a beta-lactam basically breaker, and so they break the drugs before they can actually go to work. We'll talk more about resistance mechanisms later, but that was the cell wall. Another great target is going to be transcription. So in order for the bacteria to make a protein, it has to take the gene for making that protein, transcribe it into RNA, a messenger RNA copy, and then translate that into protein. And antibiotics can target the transcription, so the making of the RNA, as well as the translation, the making of the protein. In terms of the transcription, here we're talking about things like rifampicin, which is going to target RNA polymerase, so the DNA to RNA copier. When we talk about translation, here there's a whole bunch of different antibiotics, lots of different classes, things like the macrolides, incosamides, oxazolidinones, aminoglycosides. So these are like kanamycin, streptomycin, all these things. These are targeting the bacterial ribosomes. So these are going to be the proteins that actually make the protein RNA complex that actually makes proteins. And so targeting this is either going to mess up the translation by making it so that the ribosomes can't proofread. Things like kanamycin are going to work in this way. They're going to kind of make it so that the bacterial ribosomes are misincorporating things. So this would be our aminoglycosides. And then you have things like the tetracyclines. These are basically going to prevent the bacteria from actually putting together those proteins. So it's not making the proteins, whereas in the case of aminoglycosides, it's just making really weird proteins that don't do what they're supposed to do, and then it messes up the whole bacteria. So we have these things, as well as like chloramphenicol, that's also going to inhibit the ribosome. So lots of different ribosome inhibitors, and those are targeting translation because the bacteria needs to make proteins. What else does the bacteria need to do? Well, the bacteria needs to copy its genome. So this is called replication, the copying of the DNA before the bacteria splits, so that each of the daughter cells from that parent bacteria gets a copy of the genome. This replication process, it involves unwinding the DNA in order to make copies of it. So you can use each strand as a template for making the copy of another strand, so you get 2 copies. But you have to unwind the DNA in order to do this. Enzymes called topoisomerases are going to help doing this unwinding. And if you target those topoisomerases, well, now you can then target the replication and inhibit the bacteria. And so drugs that do this include quinolones, things like ciprofloxacin. Basically, these can inhibit the topoisomerase in order to inhibit the replication. What else? We can attack the bacteria's metabolism, so they're making and breaking of molecules. So one of the molecules that they need a lot to do all sorts of things, including their replication, is going to be folic acid. And so folic acid is going to be used for making a bunch of other molecules, including DNA. And in order to make this this folic acid, you need a bunch of different enzymes, and so some of these antibiotics are actually going to target those enzymes that are needed to make folic acid. So, for example, the sulfonamides, the so-called sulfa drugs, which, by the way, were the first antibiotics to be discovered before all that mold stuff with the penicillin. Basically, these, as well as thimethoprene, these are going to target dihydroapatase synthase, which is something that the bacteria need in order to make the folic acid. And so that is another core target. So we have the cell wall, we have transcription, we have translation, we have replication, and we have metabolism. And so these are some of the key targets of antibiotics. And then we have the resistance mechanisms. So the resistance mechanisms, we can typically classify them as either affecting the drug, changing the drug, or changing the bacteria so that they can avoid the effects of the drug. When it comes to changing the drug, we can be adding things to it, or we can be breaking it down. So adding things to it, these include things like methylation or acetylation acetylation. So, for example, the chloramphenicol acetyltransferase CAT, this gene actually makes it so that the bacteria can acetylate the chloramphenicol and prevent it from binding. Similarly, there are mechanisms where they can change the target, so change the thing that the antibiotic was binding to. So you can also inhibit the effects of the chloramphenicol using the CFR gene, which methylates the ribosome at the location where the chloramphenicol binds. So you can change the drug or you can change the target. That was adding to the drug in the case of the acetylation or things like methylation or phosphorylation. So we have enzymes that phosphorylate kanamycin to prevent it from binding, say. So these are common antibiotic resistance mechanisms, as well as changing the target. So we saw there when we had the methylation of the ribosome. Now, it can be kind of hard to change, or not hard, but I mean, it can be kind of damaging to the bacteria if they change one of their key processes to prevent the drug from changing it. And so sometimes these bacteria won't make the product until they need it, or if they do make the product, they'll have a growth disadvantage unless the antibiotics are present, which is why the presence of antibiotics can help select for resistance, and why we want to make sure we're not overusing antibiotics. But those were a couple examples where we were directly changing the drug or changing the target. Another way is to kind of work around it. So if you use, say, a different protein that does the same thing as the one that's targeted but isn't targeted, that's a great way. Or if you have like kind of rescued proteins, things like the ABCF proteins, which can kind of rescue stalled ribosomes in some cases. So this allows you to kind of work around it. You can also just kind of try to keep it out. So there are ways where bacteria have intrinsic resistance. So basically, they don't need any sort of resistance gene in order to make them resistant. They're just naturally so. And so the gram-negative bacteria, they actually have two membranes. So they have like an inner membrane and an outer membrane and like a periplasmic space in between. And this, even in the outer membrane, then they have like things like lipopolysaccharides. So basically, it's an impenetrable barrier for a lot of different things, although there are channels called like porins that can let things in, and exporters that can let things out. And so those cell membranes make it a really impenetrable for a lot of different antibiotics unless they can go through those pores. So this introduces intrinsic resistance. So gram-negative bacteria are intrinsically resistant to a lot of antibiotics, which make them harder to treat in a lot of cases than the gram-positive bacteria, which only have that single thick cell membrane as well as the thicker cell wall. But in addition to that intrinsic resistance, basically even the gram-positive have a way to control what gets in and out through those porins and those exporters. And so they can change the number and the permeability of those porins, which are just like channels kind of. So you can add more if you want to let more things in, but in this case, they'll probably be adding less to let fewer things in, or changing those porins so that they don't let certain antibiotics through. Then so that's keeping it out, and you can also pump it out if it does come in. And so this includes things like the tetracycline efflux pump. You have these also like multi-drug efflux pumps, basically things that the bacteria can use in order to pump out any drug that comes in. So when we do like use tetracycline resistance in the lab as a selection marker, so though resistance is bad in like the wild, if it happens in, we can take advantage of it in the lab in order to basically select for bacteria that have a certain thing, such as the plasmid, with the gene that we want, as well as the antibiotic resistance gene. We then regrow the bacteria containing that plasmid in the presence of the antibiotics, so that we make sure that we're only feeding the bacteria that actually have what we want. So we often use that chloramphenicol resistance gene, that kanamycin resistance gene, that beta-lactamase, which is going to break down that beta-lactam, as well as the tetracycline efflux pump maker. And so these are common ways that we can use in the lab. And so you can see we're using a whole variety of these different mechanisms. And so in the case of the tetracycline, what we're doing is we're making an efflux, the cells are making an efflux pump that are actually going to pump out any of that drug that got inside. And so this is another cool way in which bacteria can get around the effects of an antibiotic by kind of just kicking it out if it comes in. Okay, so that was a basic overview of different antibiotic types, as well as different ways that bacteria can go around them. And if you want to learn more, I have much more on them in other posts and things like that. But today I just wanted to do this quick overview, get you out the door, and hopefully a little review. So I hope that helped.